Good morning, everyone. It's nice to be here. I'm going to talk about, well, the title of my talk is EBRAINS for motor neurons, network and synaptic plasticity, but of course, EBRAINS is more on the technology side. There is also a science part, so I'm going to talk also mainly on the science part, in particular, the kind of science that we do in my lab, and occasionally I will point out what you can do on EBRAINS with respect to what I'm going to talk. And I'm going to talk about modeling. In general, if you want to model a brain system or the brain itself, what you do is use the black box approach and say, okay, I'm smart enough to invent a function or an artificial network that can generalize, and I can train in such a way that to a specific set of inputs I get the right output, and I can organize things in such a way that for any input it will give me the right output. Okay? In my case, in our case, what we want to do is open the box and look inside, study the components, how they work in isolation, how they work together, how they are connected, how they evolve with time, in such a way that we can have a much more deeper information on how the box works. And I'm going to do it for a little bit for the hippocampus, which in a human has about 20 million principal neurons and is directly involved in higher brain functions, learning, spatial navigation, object recognition, and it is implicated in a lot of diseases. By the way, if you look at the picture on the right, that is a stereo view. It means that if you blur your eyes in such a way that you overlap two halves on the same place, your brain will form a three-dimensional version of it. I don't know if you can do it or not within 15, 20 seconds. If you don't, just forget it, and that will move around. In the meanwhile, like, take some water. So if you are an experimentalist, you are more familiar with the mouse hippocampus. In the human, it has a shape kind of different because it does not have the convolution and this kind of curves and sulcis that you have in the cortical area of the human brain, with respect that they don't have it in the rodents or other mammals. This is important. We will come back to that. Okay, so how do we know what we know about how the hippocampus implements cognitive functions? I'm going to show you two examples, not from my lab. This is a movie of recordings from a human of deep electrodes, and the human was shown pictures, okay? And the clicks that you are going to hear, I hope that the noise, the microphone, the clicks you can hear up to the last row, mark the activity on individual neurons in or close to the hippocampus in the presence of different pictures for this specific person at this specific time of his life. Okay, the first neuron that we're going to see is selected for Marilyn Monroe. Okay? Not only the name, but also the face, those are single units, single neurons, okay? Now, same person, another neuron. This is selected for Josh Brolin, whoever is he. I don't know him. He's an actor. This guy, of course, it is important. This one is Josh Brolin, not Marilyn Monroe, Josh Brolin. Okay? So this will give you an idea of how the hippocampus is coding the information. Those are not grandmother cells. It means that if we record from the same person 1 week or 1 year later, the same neuron may be tuned to something else. But the important information here is that information coding in the hippocampus is at the level of the single neuron. Let me give you another example. Place cells, everybody knows them. Okay? Again, those are in the hippocampus, and those are recordings from individual neurons with different colors from the right. Individual neurons code for different places in the same environment. Again, single neurons. We will come back to this kind of coding later in the talk. Okay, can we do it in a model? Yes, we do. We can. We did it several years ago. And this is an example. We have a neuron with this is a CA1 pyramidal neuron. We set up each dendrite to be selective for different features of a face, of different properties of these kind of towers. So this type of eyes is going to activate this dendrite. This kind of nose is going to activate this dendrite, and so on. Okay? Or the same with this kind of towers. And if we run the simulation, now, can you see? Yeah. So I can change here the kind of eyes. I can change the nose. It's unbelievable as the face changes with a single features change. Okay, save the code, run the simulation. Now, what we're going to do in this simulation is that at about 50 milliseconds from the start of the simulation, we are going to give the input, which means we are going to activate the 3 dendrites that are coding for the 3 features. Note that at the beginning I'm charging the neuron with the cortical input as a performance path. Eventually, and you should because I tested it, you will get an action potential in recognition of that specific face. So now what happens if I change the face? Let's say I change the mouth. If I change the mouth, now the neuron is not tuned. I mean, it is still tuned to the previous face, but now it does not receive all the features that form that face. So the neuron, it is not going to recognize that face anymore. Okay, this is a very simple proof of principle, which gives you an idea of how you can implement single object recognition from a pyramidal neuron. Let me close this and that. And that. What about place cells? We did also a model for the place cell, and how can you form a place cell without knowing which direction or which object is going to associate with the place cell. So we implement a maze, a room with 3 objects, and we let the mouse going around randomly, and at a certain point we gave a cortical input in form of the current clamp to the distal dendrite, which are known to generate plateau potentials in CA1 pyramidal neurons, especially in the distal dendrites. When you associate these 2 kind of inputs, you get the place cell. And you get the place cells oriented and tuned according to whatever it is in view at that specific time in which the performed input comes. So this is the simulation. These are the same neuron, except that in this, the same path, except that at this point in time, a reward is coming while the virtual mouse is looking at this object or at that object. So it's the same neuron, but you can tune it to 2 different objects according to when you get the reward. Okay, let's move on. Once that we have this kind of proof of principle, we can go on and try to build the circuit. So we went to the experimental lab, in this case UCL, and we collect several CA1 pyramidal neurons. We wanted to, in this case, we are interested in implementing a CA1 region, which makes up to 1/2 of the hippocampus. So pyramidal cells, interneurons, and you see that here you see both the dendrites, the soma, but also the axon. And I think that this will come back to the question that was raised before. If you look at the axon from the green interneuron here, this is an OLM, interneuron. So you know that the dendrites are in the corresponding to the basal dendrites of the CN1 pyramidal neuron, but the axon goes back and is extensively developed in the stratum lacunosum molecular. Okay, so you want to take into account this kind of non-uniform, non-isotropic connectivity when you build your circuit. One way to do it is to use a touch detection bottom-up approach, in which you have the reconstructions of both the dendrites and the axon. You put everything together, you distribute your neurons in the volume determined by the atlas, and you build the connectivity. It will automatically come up as it is in the real brain. Or you use a mathematical algorithmic approach in which you can develop and generate the same connectivity that you see in brain regions, but you don't, or if you do at your own risk. You don't do a connectivity at random using an isotropic connection, maybe an exponential random connectivity, because this is significantly, I would say statistically different from what the brain region is connected with. Okay, so you need to be careful when you build your network. Once you have this, of course, you need to generate models of the type of traces that you record. So we used the 1500 experimental traces. We considered over 100 electrophysiological features, spike amplitude, duration, spike times over different currents, and interspike intervals, bursting properties, and resting potential, and so on. And we can calculate this automatically using, and this is one of the keys that we are going to see today, using EBRAINS. So this is, for example, the neural extract features. You can go on. This is very simple web app in which you can select, for example, this from Thomson Lab, hippocampus, CO1, let's say the pyramidal cells. And so, but you can use, upload also your own traces. You can mix your traces with the other traces that we have in the knowledge graph of the EBRAINS, put them together and calculate the features. In this case, you can apply, get the traces. Okay, you see traces here. You can select all of them. I just select a few of them, and then you can extract the features, agreeing to whatever. You know, you usually click on agree, but then you're in trouble, okay? But in this case, on EBRAINS, you are safe. And then you can select a bunch of shape, spike, events, or voltage features. There are hundreds of them, okay? You don't want to use all of them because you are, again, it means that you don't know what you are doing. You want to select the one that you want to use for your model, okay? And then you can get them, I mean, I just go on, and I don't want to take too much time. But this is very easy web application that you can do to begin to build your own model. We did that, and we get, of course, the model that you can compare with experiments. It is very close if you look at the average. Also, it is relatively also good that we can reproduce in some way the variability of the experimental data. Again, with these kind of models, you can build it automatically without downloading anything, okay? You just do everything online. You use the Hodgkin-Huxley neuron builder that you find in the workflow and use cases for cellular level simulation in EBRAINS. There are a bunch of them doing trace analysis, single cell modeling, morphology, in silico experiment, and so on. In this case, single cell modeling, you have the Hodgkin-Huxley neurobuilder. And this gives me the opportunity to give you a more specific information on what you are going to see on EBRAINS. Because, for example, you see a use case, you may say, okay, I'm going to use it. Well, you need to be careful again, because you know, in some cases you need to know, you need to know what you are doing. And this kind of icons here will give you more information. This means that everybody can run this without problems. This means that it is organized as a web application, so you just point and click. There is even an interactive video tutorial that teaches you how to go through the use case in this case. And in particular, this one on the automatic optimization of a neural model from experimental data uses HPC, a supercomputer, to help you in doing whatever you want to do. And let me see if I can, I'm just faking it, but I just want to show one particular interesting feature of this thing, because once you get the features, you can set up the optimization. You can run the optimization by point and click. You have a set of different supercomputers that you can use to that purpose. At this point, there is the CSCS, which is in Zurich. You can have your own account, and then you use your account, but you can do it through the service account, which means that we give you free supercomputing time, not a lot of it, of course, but free supercomputing account in which you can launch your optimization without doing anything, not even an account. You just go there, organize, set up your files, and launch the optimization. You can set up your model. Okay, let's close this. Go on. Of course, you have your model, but you need the synapses connection. Otherwise, you cannot connect the network. So we went through the, I would say, painful work to go through all the possible interconnections between pyramidal and the plethora of interneurons that are in the hippocampus, and we fit every single one of them. Again, you should be aware that you need to know what you are doing. Again, you can do this on EBRAINS. There is another use case. This is trace analysis. You see here that the research instruction of synaptic fittings event. Of course, in this case, you must be a power user, which means power user means that you know what you are doing with coding. In fact, it is not a web application anymore, but it is a Jupyter notebook. Okay? But you can do it automatically. You can get your free time on supercomputing before getting your own, and so on. Okay, so. Of course, this cannot be done by a single lab, so we call ourselves the HBP Hippocampus team, and there is we are collaborating with the Blue Brain Project, the UCL, the group in Budapest, and finally we get, this is the current version of the full hippocampus CA1 region of rat. It is 450,000 neurons, 90 million of membrane segments, a bunch of equations. Of course, we run it on Phoenix supercomputing, and this is an idea of how the system looks like when you launch it, put everything together, and let it run. This is a preliminary version, and there is not much inhibition. As a matter of fact, this version has not been validated against experimental data. If you put all the experimental data, which means whatever you know about the hippocampus at this point, what you get is a stone-cold system. You don't see this kind of traveling waves. Okay? These traveling waves you see when you take out inhibition or reduce inhibition, which may be a pathological condition. But I think I can show you what is the current version. Of course, at this point we can do in silico experiments, okay? And in silico experiments means that you have the system, and you can begin to do again through EBRAINS. You go here to the brain area in silico experiments, hippocampus. Oh no, this is to do the simulation. I don't want to do the simulation. I want to do the recordings. I have the system here, I have all the cells. I can select the cells from which I want to record. Let's pick up, this is a CA1, okay? This is an interneuron, okay? And then I can say, okay, I want to all the afferents, all neuron this, add all the afferents or the efferents of this neuron, and add filter, and I have the connectivity that I choose. At that point, I can set up the recordings. And let's put the recordings. Simulation config. Okay, proceed to simulation. Let's do this. Five minutes. Okay, good. Okay, so here. What? 5 minutes. Okay. Okay, so you want me to use more? No, I don't. I'm not. It's going to be rude. I will stop in 5 minutes. So I can do the in silico experiment, okay? And of course, the idea is to connect it to everything and to do the closed loop simulations. And of course, if I have the entire hippocampus, I can do my robot doing everything I want. But we don't need the entire hippocampus. I showed you before that we need single cell. Okay? So the idea was, suppose that you are coming here for the first time. You, of course, need some instructions to come to this place. But if you come back tomorrow, you don't need a lot of training like an artificial intelligence network will need. You just come back, okay? As a human, we do navigation very efficiently, okay? So it is clear that there is something that is missing from the way in which artificial intelligence networks do their job of finding the right path in a maze. And so we try to do it using hippocampal circuitry. So what we did is to build a very simple network, about 30 neurons, implemented in Nest, actually in the Pine, with space-time dependent plasticity. There are place cells, object recognition cells, persistent firing, no interneuron at this point, plastic synapses. We connected them to a robot. Actually, the code is in the robotic platform of EBRAINS. And we connect to a robot through a ROS Pine interface. ROS is the acronym for Robot Operating System. And so we connect to get the data, to have motor control, and this is what happens the very first time. And we let the network evolve with time as the robot navigates and look at different monitors of different colors. This is, of course, a proof of principle with a lot of limitations. So it is learning. There is the red, the blue, then I see the black, and then I see the exit. Well, this concludes the learning phase. No trials, no huge amount of trials to go after or every possible sequence. This is only one sequence, only one training event. And then this is what happens during the call. Okay? Single training, single, and then you get the learning. Even if you put walls, because you have this avoidance. Okay? If you put interneurons, a few numbers of interneurons, like the 10% that there is in the hippocampal circuitry, you connect this, of course, using your intuition. What you get is that the network now is able to let the robot navigate a four-armed maze, going through different paths, forgetting the wrong paths, and going to the correct path in a single learning phase. So I'm going to show you this. The robot is going to go first to this wrong path, goes back, and then takes the new one. I'm telling you because this is kind of Formula 1 stuff, so it may be difficult to figure out. It is going through this path, then it figures out that it is wrong because of this object signals a wrong end, goes back to the initial, then goes back here, forget about this path, choose another one by random, which is the correct one, and this will conclude the learning phase. Okay? During the recall, it goes directly in the correct branch. Okay. If you look at the pattern of the entire network, you will see the usual pattern that you see in the experiments. However, during the recall, if you reorganize as they do in experiments with the firing of the different neurons, you see the sequence just like in the experiments. And now we are improving the network, removing some limitation, and of course porting the network to autonomous real robots, so we have no Wi-Fi control from an external laptop. And especially we want to do learning and recall during navigation, because this is what we humans do. Okay? We don't do a learning phase and then the recall phase. We do learning and recall as we navigate. Okay, so I talk about that. If you are unsure about how to use EBRAINS, we do, I need to skip the hippocampus out because there is no time, and this is part of my lab that has been involved with this. Thank you very much.